for having me in this conference. So I'm going to talk about a family of protein, the TIM family of protein, and how X-ray reflectivity as one of the three methods allow us to really hone in on the differences of this family of proteins in terms of their interactions with the membrane surface. So just a little bit of the background. We've heard earlier about sort of asymmetry in the cell membrane, and it is in the cell membrane an active process to keep this asymmetry. So what is shown in this cartoon is that the extracellular version on the outer leaflet of the membrane is normally sort of spheroionic in nature, and the charge are being really actively controlled to be flipped on the inside, the inner leaflet of the membrane. And there are cases when membrane exposure with phosphorylserine is important, and there's a bunch of biological process that would lead to this PS exposure. One of which is the apoptosis, in which case that, when cell death is undergoing, it is not possible to have enough energy to really keep this asymmetry in the correct way. And as a result, over time, PS is being exposed. Now, over the past decade and a half, a lot of work has been done in looking at the PS recognizing protein. And I'm just highlighting just two of them. We're going to talk about the TIM family of protein, which is important for immune regulation today. This is a calcium-mediated interactions, whereas the MFG8, the mil-fat globulin protein, which is also important for apoptosis, is actually a non-calcium mediated event. And there is a whole slew of other proteins that are recognizing on the PS. So if I were to look at this TIM family protein, which is a transmembrane as seen in the structure, immunoglobulin and mucin, it has a long mucin domain that is there, TIM protein that is shown in the cartoon here. The three family members, TIM 1, 3, and 4 are shown. And if we were to focus on the binding domain, they all have a single calcium-dependent PS binding site on the surface, as shown this overlap of the structure of the binding domain. And the functions of TIM4 and TIM1 seems to be somewhat well understood. TIM4 is the receptor for the PS when the cell undergoing apoptosis, so that it is a clearance mechanism that you can recognize the cell is undergoing cell death. And TIM1 seems to be an immune stimulator. And what is less clear is TIM3. What is the function of it? Well, it is thought to be an immunosuppressor, and a lot of work has been done to look at TIM3 as a target cancer immunotherapy. And TIM3 interact with an array of receptors has been identified with these list of ligands that are there, including the PS that is shown highlighted in this. So we'll just focus on the PS. And just to sort of remind you, all of these have a single binding pocket. And on the single binding pocket, it also have a tryptophan that is there. And as you can clearly see in the TIM1 case, you really see that in the crystal structure, the binding domain is actually close, and that becomes important later on in the talk. And you see that TIM3 and TIM4. So the question we want to ask is the density of the PS that is on the surface being sensitive? When we just choose one of the TIM family members, TIM4 for the purpose of illustration. And it turns out that their sensitivity of each of the TIM family is a little bit different. And this gives rise partially to the selectivity of the differential recognition of different membrane surface when they have to sort of upregulate, downregulate immune response, or for clearance purposes. So the first type of assay that we are going to do to get at the binding is a fluorescence spectral shift that we're looking for when the protein is free versus when the protein is bound. And so it is a calcium-mediated surface, and we used last unimellar vesicle as a model system for the bilayer, and we control the amount of PS, which is represented in the cartoon in this red dots there, to change the surface density and the surface presentation of that. And so what you see is that between the unbound state, whereby the tryptophan is in an aqueous environment, versus in the bound state, whereas it is inserted in a hydrophobic environment, the spectral shift is occurred. And when we were to look at the changing the mole fraction of the PS that is being presented, which is on the x-axis, you really see a sigmoidal curve that can fit to the Hill model. And the Hill model fit gives us a Hill coefficient of a number larger than 2, which suggests that it needs at least 3 PS to enhance for the stable binding of the protein to the surface. And we can go through the exact sort of association of the surface. But if you were to look at, and later on I'll show you the technique of looking at its embedding into the lipid domain, the environment of the tryptophan that is being changed is sort of depicted in the picture on the right-hand side. And so it turns out that one can sort of, change the PS concentration, but I won't be able to go into the detail, but the equivalence of changing the PS surface mole fraction can actually be brought about by tuning the quintessential binding partner, that is the calcium, that is important for initiating a change. And as you can see in the curve on the right, what you can see is that you can change the calcium concentration that is being presented on in the experiment, and you see a corresponding shift on the spectral shift, also indicating that it tuned the amount of surface penetration and binding of the protein into the membrane surface. So then the question is that, okay, if it is sensitive, which we have seen by the spectral shift and the degree of the shift, how does the PIM4 with a single binding pocket be suggested by the Hill coefficient that it has multiple PS binding partner? And so to get at this question, we want to look at the structure of the protein, not so much by itself, but when it is being bound to the membrane surface. So X-ray reflectivity using a monolayer system, model system, is being used to decipher the structure of the protein as it gets to bind with the lipid membrane. And we put it at a bilayer equivalent pressure. And I don't have to explain to this group that there are parameters that you can get out from in terms of the electron density, as well as the length of the different blocks that is interacting. And one can get the reflectivity curve that is there. So fitting would give you, in the presence of TIM, this bump that you see at below the head group area of the protein ad layer that is there. And what we have used is to just show that if we were to remove the calcium by EGTA, then there's no difference. Or if we were to remove the PS, which is the binding partner, that there's also no equivalent ad layer that is absorbed at the interface. And so the X-ray reflectivity analysis that we do in the presence of the protein, use the structure of the protein as a starting place. And what we do in order to see the orientation is to sort of systematically rotate the protein, generate what would be the computational version of what the reflectivity would be like by projecting its electron density to the z-axis. And if you were to rotate it and look at all possible conformation, each time comparing with the experimental result, you would get something like the heat map that is shown here, that if you were to rotate in the phi and the theta directions, you would see that there are pockets whereby you have the highest correlation or highest match to the X-ray experimental structure that we obtain. And so in this particular case, you see that there are two possible wells in which that there are likely protein orientation at the surface of the protein. But if you were to go back and look at the X-ray scattering details, really the fit doesn't show any difference. And the cartoon below in the A and the B configurations show how the protein would be like. But if you were to recall that this is a mucin domain that the protein has, that the part that is tethered to the mucin in the B configuration is not possible to be binding to the lipid surface in this configuration. So that allow us to eliminate the orientation of B as a possible orientation of the protein interaction. So with X-ray reflectivity, we were able to get at the interactions that is consistent with the known features of the protein by using this sort of systematic alteration of the angle of orientation of the protein to the interface, whereby you see the central head group of the PS position and the hydrophobic loop penetration, which is in line with the spectral shift that we have observed in the other experiment that I showed you earlier. But the reflectivity itself, as you see, cannot really resolve in the blob the more atomistic detail of how the various portion of the protein that is in close vicinity of the surface interact with the lipid. So in order to get at that, we move to molecular dynamic simulation that allow us to get at the detail. And just going through the results quickly, what you see is that the central position is in line with the membrane phosphate, and the hydrophobic penetration is in line with the shift of the spectral shift that we observe. And the orientation between the experimental fit versus the simulated fit by the rotation that we explained earlier are also in line with one another. And what these results allow me to look at is really in close proximity of the membrane surface. What are the other players that are at hand that give rise to the Hill coefficient of a number that is larger than 3? Knowing that there is a single binding site for the PS, which is mediated by calcium in the middle. And what we found is that there are positively charged lipid amino acid that is close to the lipid acid, one in this orientation, and then the other one in a perpendicular orientation. That is important to stabilize the interaction of the peripheral binding protein into the lipid membrane layer, so that the number of 3 minimum charged lipid that is necessary for a stable configuration of binding at the surface is really being, as this would suggest, mediated by these positively charged amino acid at the interface. So in order to really find out whether they really contribute to this binding and whether there are additional PS specificity in this, we move on to do a aniline mutant binding assay, whereby by single site, we remove each of this positively charged amino acid and look at the effect on the ability of the protein to bind to the surface. And in all but one case, which is within error, you really do see that the decrease in the propensity of the binding when even one of these 4 peripheral sites are actually mutated and replaced by the aniline in the system. And so the story that I've told you thus far pertains only to the TIM4 protein. But then there's TIM1 and TIM3, and do they have the sensitivity to the PS surface density as the cousin TIM4 as shown earlier? And so we try to look at using the similar type of binding assay with the TIM4, and this is just to remind you what you see earlier, that you have a response that is a sigmoidal curve, and juxtaposing this are the result of the TIM3 in this curve, which you see a much stronger binding, and TIM3 in this light gray curve, which has a much lower affinity to the surface from the assay itself. And the S curve that correspond to, as I said earlier, the TIM4 have multiple PS binding partner, and the other 2, would they be similar? And you can fit the sigmoidal curve, and they have different numbers from the Hill coefficient that you obtain from TIM4. But the issue is that TIM3 in this particular case binds so weakly that makes it very difficult to produce a similar type of accuracy when we do the same X-ray reflectivity measurements and rotation on the surface. And if you recall, in the case of the TIM1, the binding pocket in the crystal structure is actually closed, making it also impossible with this iterative process to match to the correct orientation of the binding of the TIM1 to the surface. So in order to sort of rectify these differences, what we did are two different things for the two different proteins. This is just a picture showing the colocalized PS head group with the binding pocket of TIM4 open, which is not the case for the TIM1. And in order to really increase the accuracy of our third set of data with the simulation, what we actually do is that we try to open up this pocket by doing first calcium equilibration, and then we do equilibration of the system into the membrane and dock it on the surface. And when we do that, all of a sudden, we're able to really have much more meaningful data and results with the equilibrated structure on the surface and decipher what is the orientation of the protein of the surface. So on the left, what is shown is the calcium equilibrated surface, and on the right is the membrane equilibrated surface. And when we want to run using a coarse grain method, the highly mobile membrane mimetic method, to really stimulate now this open pocket is binding to the surface, what we found is that the bound orientation is very much similar to the membrane equilibrated position that allow us to really get at the orientation of this TIM1 protein onto the surface. So with that, we were able to get at the interactions of the TIM1 and the orientation of the surface. And now we need to sort of, in order to have this comparison, we need to find out a way of understanding why TIM3 is binding so much weaker. And so there are a couple of reasons for that. First, in this look at the comparison of the penetration, you can see that TIM3, which is on the right, needs a much greater footprint for the membrane penetration. And therefore, under the same lipid condition, the binding is weaker. And so this is bared out because if you were to move it to more unsaturated lipid tail, then you were able, in the case of this curve, get a much stronger binding signal at a lower calcium concentration in the substrate. The second reason is that TIM has actually a lower calcium affinity to the surface. And we can actually see it by this sort of KD curve of the calcium as a function of the three different proteins. And so we have seen that we have the PS specificity that I argued earlier on, because in the real life cases, PS is used. But if the other two lipid that is necessary to stabilize the surface binding of the protein, is PS necessary, or can PS be sort of switched out by other lipids on the surface that has that anionic specificity? So this data is quite dense, but what I'm trying to show you here is that with TIM4, if I were to replace part of the PS by PG, so just keeping 10% of it being PS with 20% of it being PG, you actually reproduce a very similar type of binding profile. But if I were to remove PS altogether, you get very, very low binding. And this is just another way of looking at it with the PG as a function of the PS mole fraction that is onto the surface. And you can play out in terms of replacing the percentage of the PS on the surface by the PG so that the total percentage will remain the same, and you get very similar type of sigmoidal type of work. But on the other hand, instead of having PG, you have a different head group PA. You already see that there is drastic difference in the case when you have complete absence of the mole fraction of PS in the total 30% mole fraction. So to sort of make the long story short, what has been discovered in this PS replacement is that depending on which protein that you're looking at, some of the protein, in the case of TIM3, we can highly enhance its affinity and binding to the lipid surface layer by replacing a small percentage of the 30% charge by a different lipid with an anionic specificity. However, the anionic specificity, depending on the head group configuration, can have a drastic effect. What is shown here in this curve with the square is the case when you replace a small percentage of PS by PA. And you can see that the KD dropped drastically, even with a 1% replacement of the PS by PA. But if you switch to a equally anionic lipid of PG, what you can see is that a much muted type of response in terms of this replacement. Even at a relatively high change of 10% replacement, you never see the drastic fourfold changes in the affinity of the surface. And it turns out from the immunological perspective, this is really important because of the small amount, less than 1% of PA being presented. And that PA actually tuned the surface sensitivity of the binding of the TIM protein, in this case particularly, TIM3 association with the lipid surface. So one can imagine having all these TIM family protein being on the surface of the cell, patrolling on the surface, and under different conditions, TIM3 would never be able to see a PS-rich protein because TIM1 is so strong. But when the surface contacts of the lipid is changed, this can actually outcompete binding of TIM1 or can co-bind with equal affinity of the TIM1 to the surface, which points to the importance of the surface regulation of the lipid presentations to the biological sensing and recognition of the surface that leads to important downstream event. So to conclude, I hope I have introduced you to this family of protein, all of which have very different binding characteristics. And the TIM can actually use the lipid contact to differentiate which one should be binding for different recognition purposes. And the methodology that presented here, using a combination of fluorescence, X-ray reflectivity, and molecular dynamic simulation, allow us to overcome some of the difficulty of studying membrane recognitions as the protein is binding to the surface. And this approach, we show you an example of the TIM protein, but it could be broadly applicable to other peripheral binding protein. And so with that, I just want to thank the people who really did the work. Most of the work is really done by Daniel Kerr, who worked on the protein system with a project that is started by Greg Tidgen. And with that, I think my time is up. And thank you. Happy to answer any questions.